Okay, hello everybody. So this is another podcast in the series from the Molecular Genetics module, Year 2 Biomedical Sciences at the University of Bradford. And for the next 45 minutes or so, we're going to be talking about regulatory RNA. So the GIF image that we see here on the beginning of the slide is an RNA molecule. The most notable aspect of RNA obviously is that it's generally single-stranded, so you don't have the classical double helix structure we observe for DNA, although obviously the base composition of RNA is very similar to deoxyribonucleic acid to DNA. Regulatory RNAs are a relatively new field in some ways and an ancient field in others, and we're going to be discussing some of the classical examples of regulatory RNA and also considering some of the more recent additions to this growing catalogue of molecules. And I want to try and explain really, I guess, in a nutshell, why this is such an important and interesting emerging area of molecular genetics. So over the course of the lecture then, we're going to be initially just considering the thing that kind of kick-started the concept that RNA might be important in a regulatory capacity, and that's the so-called RNA World Hypothesis that was postulated 50 or 60 years ago now by Francis Crick. Before launching into an overview really of the different classical non-coding RNAs that exist, or regulatory RNAs as they're also known, that exist in the cell. As I mentioned, this is going to include a discussion around some of the classical examples of non-coding regulatory RNAs and also some information about the more recently identified and, if some people would argue, more interesting examples of this family of regulatory molecules, and then we'll finish by summarising the lecture. So the learning objectives, well, they look brief, but they're going to require a reasonable amount of work on your part, and that is to understand, broadly, conceptually understand that non-coding RNAs are key regulatory players in eukaryotic cells. So I'll explain in more detail what I mean by non-coding RNAs later on, but essentially these are RNAs which aren't mRNA. So as you should be aware by now, it is messenger RNA, mRNA, that is the product of gene transcription, and it's these messenger RNAs that contain the information that's used by the translational machinery in order to produce then gene products or proteins. There is, however, an entire separate, if you like, family of RNAs that are transcribed within the cell which don't go on to make protein but nevertheless are incredibly important for cellular function, and that's what we're going to be considering today, these non-coding RNAs. So you need to understand that conceptually, and also you need to be able to describe examples of these non-coding RNAs, particularly the ones covered in the lecture, and you need to be able to discuss how they operate, so how these non-coding RNAs function within the cell in order to allow the cell to regulate primarily, as we'll see, gene expression. So then, this so-called RNA World Hypothesis, well, what is it? Well, this is an idea that was proposed initially by several scientists. Often Crick is the guy that gets the plaudits for proposing this hypothesis, but there were other people involved in postulating its importance as well, including Woese. And this all happened around the 1960s. And in a nutshell, what these guys were claiming was that early life on the planet actually began as a self-replicating RNA molecule. So this is to say that the first origins of life and the primordial soup were not proteins, as many people had originally suspected. This concept that there may have been a formation of a polypeptide of individual amino acids coming together to form a polypeptide, and that was the first actual signs of life being created, if you like, on the planet. What these guys argued was that first actual biological molecule was more likely to be RNA. So when they made this statement, it was quite controversial because at that time in the 60s, certainly the vast majority of the scientific communities believed that proteins were the only molecules capable of catalytic activity. So in order for enzymic processes to occur, you need a molecule that can perform catalysis of other molecules, and up until, actually the 1980s, it was widely believed that only proteins were capable of performing such a function. However, 20 or so years after this RNA World Hypothesis was put forward by Crick, him and his fellow believers were proved to be, well, the jury's still out, but certainly the evidence in support of their hypothesis gained significant ground, following the discovery of these so-called catalytic RNA molecules, or ribozymes. So ribozymes are effectively RNA molecules which can, in all intents and purposes, function like a protein. So they're capable of possessing catalytic activity, self-catalysis of RNA sequence, and also modification of other things as well, proteins, etc. Perhaps the best example of a ribozyme that you guys will be aware of is the actual ribosome itself, so the machinery that translates messenger RNA sequence into protein. So if you look here at this image on this slide, this is an actual computer generated image based on crystallography data of the ribosome, the large and small subunit of the ribosome here. The green components of what you are seeing here are the protein components of the ribosome. The blue components are actually ribosomal RNA. And if you were to take away the protein elements of the ribosome, what you would be left with is a structure that's actually very reminiscent. In fact, it is more or less an intact representation of what the ribosome looks like structurally. And what that kind of tells us is that it's RNA really that's responsible for much of the structure and certainly much of the function of the ribosome with regards to its role in protein translational machinery and the production of proteins. And it's further evidence that RNAs actually are very important molecules with regards to enzymic activity within cells. So this concept then of regulatory RNAs, how were they first actually discovered? What was the first regulatory RNA to be identified and described? Well, it's this guy again, Francis Crick. He's primarily famous, obviously, Francis Crick, for his work with James Watson on the structure of DNA, but he did really, go on to produce some other very important seminal pieces of work for molecular genetics and biology in general. You guys, I believe, may have done the Crick paper in year one, looking at the identification or description of the genetic code. That was another one of his major pieces of work and relates to this discovery of the first regulatory RNA. So it wasn't Crick actually identified the first regulatory RNA, but he was involved in postulating what it might be. So during his paper on the genetic code, where Crick effectively works out that the information contained within DNA exists in triplet codons, he postulated that that information was likely to utilise some kind of adapter molecule prior to it being translated into actual protein sequence, into amino acid sequence. And he wasn't explicit about what that adapter molecule was, but he did postulate in his article that it was likely to be RNA. And once again, Crick was proven to be correct. Several years later, in fact over a decade later, work by another scientist called Holley identified the very first, if you like, regulatory RNA molecule, which was the tRNA molecule for alanine. And we've got an image of it here. It's this classical cloverleaf structure, and this is an actual, if you like, a computer-generated image based on NMR crystallography, NMR studies and crystallography from the tRNA molecule. And what Holley found was that this tRNA fit the bill with regards to being the adapter molecule that Crick had hypothesized about over a decade earlier. So the first regulatory RNA had been identified, and from there on in, really, the entire field started to gather pace. So if we think about the content of a cell, the nucleic acid content of a cell, and in particular we think about the RNA content of a cell, as undergraduates learning this field for the first time, and you will be forgiven for thinking perhaps that the most abundant RNA that you would expect to find in a cell would be mRNA. After all, one of the major purposes of the RNA molecule is to convey the information contained within the DNA sequence to the translational machinery so we can make proteins. So it seems logical, therefore, that the cell will be producing a lot of these mRNAs in order to fulfill that role. In actual fact, if you look at total RNA from a eukaryotic cell, a human cell, and ask which RNA species is the most abundant, by far the most abundant RNA species that you will find within the cell is ribosomal RNA. So these are RNA molecules which comprise the ribosomes that we looked at a slide or two ago. And these ribosomal RNA genes are interesting in themselves as they're found as tandem repeats across the genome on several chromosomes, 13, 14, 15, 21 and 22. So the diagram here basically shows how the ribosomal RNA gene, if you like, so the ribosomal DNA exists within the cell nucleus. Now, these genes are actually found in the nucleoli of cells. So remember, the nucleoli are these dense subnuclear bodies, which are often termed ribosome factories, for the simple reason it is where the ribosomal DNA and RNA is produced in order to then assemble mature ribosomes, which are then exported out into the cytoplasm and go about the business of making protein. So as we can see here, ribosomal DNA exists as this this transcription unit here, which is comprised of different ribosomal DNA genes: the 18S gene, the 5.8S gene, and the 25S gene. And this is transcribed and then processed in such a way that you get assembly of these different ribosomal RNA molecules into what we would then recognize as the mature ribosome molecule. In eukaryotic cells, the complete ribosome is termed 80S. Remember, in prokaryotic cells it's termed 70S. In eukaryotic cells it's slightly different, the structure of the ribosome, and it's termed 80S. And of this 80S ribosome, we can break it down into two further subunits: the large 60S subunit and the small 40S subunit. Now, each of these two smaller subunits themselves contains different versions of the ribosomal RNA. The 60S subunit contains a 5S, the 5.8S, and the 28S ribosomal RNA, whereas the small subunit, the 40S ribosomal subunit, contains the 18S ribosomal RNA molecule. Both these subunits are processed and produced in a quite complex manner that occurs co-transcriptionally. So whilst the ribosomal DNA has been been transcribed, you will notice here that the 5S, which is found in the 60S ribosome, isn't present in this transcription unit, and that's because the 5S ribosomal RNA gene is actually located in a different tandem repeat elsewhere in the genome. But it's similar to this multi-gene repeat that we have here, this transcription unit, insofar as it's found in multiple tandem copies. It's interesting to note that these subtle differences in ribosome structure have been exploited by pharmaceutical companies to develop some of the more potent antibiotics actually that we use now in the clinics. For example, chloramphenicol, erythromycin, streptomycin, all of these antibiotics work by virtue of the fact that prokaryotic ribosomes differ slightly to eukaryotic ribosomes. So these antibiotics recognize and interfere and inactivate 70S prokaryotic ribosomal subunits, but not the 80S eukaryotic ribosomal subunits. That's why you can take chloramphenicol, it won't actually damage your cells, the eukaryotic cells, but it will interfere with prokaryotic bacterial translation and therefore kill bacterial infection. So I've alluded to ribosomal RNA processing and the process by which ribosomes are constructed. We don't need to worry, or you don't need to worry, about the detail behind ribosome assembly. That's something beyond the scope of this lecture. I do need you to understand the fact that the ribosomal RNAs are structured in these tandem repeats and these transcriptional units. You do need to know that. You don't need to worry about the full process of how the ribosomal RNA is assembled into ribosomes. But it is important that you understand some of the other players that are involved in that process, because one of them is another key regulatory RNA molecule, and that is this so-called small nucleolar RNA, or snRNA. So what snRNAs are a specialised type of regulatory RNA that are involved in this ribosomal RNA processing procedure. Now, they're usually encoded within the introns of genes which encode ribosomal proteins, and the snRibosomal RNAs, the snRNAs themselves are also transcribed like messenger RNAs by RNA polymerase II. So let me just explain and reiterate what I mean by that point. If you think about the genome, we have numerous regions of the genome which we know contain coding regions. So these are regions which are going to express a gene which will go on to produce a functional protein product. Yeah? Those protein products may include some of the proteins which are actually used to construct the ribosome, so ribosomal proteins. Now these small nucleolar RNAs, these are actually transcribed from the so-called junk DNA, which is found in the intronic sequence of these ribosomal RNA genes, of these ribosomal protein genes. So if you imagine a ribosomal protein, a gene which encodes a ribosomal protein as having exonic regions and then an intronic region, an exonic region and an intronic region. Obviously these intronic regions will be removed during mRNA processing to leave us with just a mature mRNA, which will then produce a ribosomal protein. But it's naive of us to think that these intronic regions are doing nothing if they're just present as junk in the actual genome, because it's within these intronic regions that these snRNA sequences actually reside, and it's from those intronic regions that RNA polymerase II copies these snRNAs to produce this small regulatory RNA molecule, which then itself is involved in processing ribosomal RNA. So what I'm trying to paint a picture of here is, and to lay the foundation for, is this understanding that the human genome is very complex, and whilst only a very small proportion, as we'll talk about later on, is actually used to code for proteins, the rest of it is not junk. Yeah. Up until very recently, a lot of scientists would have described it as junk, but it's now naive and incorrect to think of it in that way. So it seems that of these sequences which don't make proteins, there are actually a hell of a lot of them are involved in making other things, which are then involved in controlling the expression of protein production. And we'll talk more about that later on in the lecture. So getting back to these snRNAs then, which are produced from the introns of these ribosomal protein genes, what they actually do effectively is they recognise and bind to ribosomal RNA and modify it in such a way that it can then go about its business and contribute to the construction of a mature ribosome. There are two flavours of snRNAs. There's a family which are called CD box snRNAs, and then another family called H/ACA box snRNAs. The CD box snRNAs methylate ribosomal RNA. The other type, the ACA box, do something called pseudouridylation of a ribosomal RNA. In effect, what they both do is they modify the actual bases within the ribosomal RNA sequence, one via methylation, one via the addition of uridylation groups. Yeah. Again, at this level, you don't need to worry too much about what those modifications are doing. What I want you to appreciate is the fact that these snRNAs exist, where they come from, and that they're involved in the modification of ribosomal RNA in such a way that the ribosomal RNA can then function correctly in its prescribed role in ribosome assembly. So another small nuclear RNA, another example of a small RNA is the so-called small nuclear RNAs, or snRNAs, not to be confused with snRNAs. So I apologise for the nomenclature. I know that genetics can be a pain in the neck with all the different protein names and gene names, etc., and it is easy to confuse these things, but try and keep the things separate in your mind. So snRNAs, small nucleolar involved in ribosome biogenesis, etc. snRNAs, small nuclear RNAs, and they're actually performing very, very different functions in the cell. So what small nuclear RNAs are actually involved in is the regulation of splicing. So we talked briefly in a previous lecture about gene expression, about how the removal of introns is carried out by a process called splicing. We mentioned the spliceosome as a very complex multi-protein machine. We didn't go into much detail about how that machine works. We're going to cover that next year in a lot of detail. But I do want to talk a little bit now about that spliceosomal machinery, because it's within that machinery that the small nuclear RNAs reside. So these small nuclear RNAs, what they do is they actually associate with over 300 other proteins to form this so-called spliceosome. And a spliceosome is one of the most complex macromolecular machines you're going to find in the cell, and its job is to recognise and remove this non-coding intronic sequence from genes which are going to go on to produce mature mRNA and protein products. So how these actual small nuclear RNAs facilitate that is it's their job to recognise where an exon ends and an intron begins in a gene sequence. So remember, if you think about a eukaryotic gene, it's comprised of several exons and several introns. Those introns need to be removed, but I don't know if you've ever considered or thought about how the cell exactly recognises where the exon ends and the intron begins. in order to accurately remove the introns without taking any of the important coding information away as well? Well, the answer to that question is that it's achieved via the function of these small nuclear RNAs, which recognise the exon-intron boundary. And we'll go into more detail about how that's recognised next year. But I want you to understand that that's what these molecules do at this point. They're also very important in a phenomenon known as alternative splicing, which you've encountered on a number of occasions. I mentioned it earlier on in this module. It's also mentioned in the human genetics, and this is a concept whereby higher order organisms such as ourselves are able to utilise a protein coding gene in more than one way. So that's to say that just because we express a single gene, that doesn't mean that single gene will just make a single protein. And it's this phenomenon of alternative splicing really that is one of the ways in which we can explain how humans can be so complex as an organism but actually contain so few genes. So, as you've been told before, we have a similar number of genes to this model organism C. elegans, which we'll talk about in the next lecture, yet obviously we're far, far more complex as an organism. And one of the explanations about how that can arise, how that's possible, is via the phenomenon of alternative splicing. The fact that of these 20,000 genes that we express, we are able to produce numerous variations from each of those genes via shuffling and using the information present in exons in different ways, by alternatively splicing those exons together. So I don't want to get into any more detail than that at this point. It's something we'll talk about in year 3, but you need to be aware that these small nuclear RNAs are integral in that process and form part of the splicing machinery. So just a quick recap then on what we've talked about so far. So the RNAs that I've discussed thus far, what we could consider really the classical regulatory RNAs. We've known about these for a reasonable number of years now, 20 or 30 years in some cases. And they all kind of support this initial hypothesis which was proposed by Crick and others about RNA World. It being an RNA world, RNA being the regulatory molecule that was first arose in the primordial soup. We've talked about ribosomal RNA assembly and the role that the small nucleolar RNAs play in that process, and we've also talked briefly about these small nuclear RNAs and their role in splicing and the regulation of alternative splicing. So remember, these small nuclear RNAs are the molecules which allow the spliceosome to correctly identify where an exon ends and an intron begins. So I want to talk about for the rest of the lecture is this new wave of RNA, regulatory RNA molecules, which really have only come to the forefront of science in some cases, the last 2 or 3 years. So this is a really new area. This is a new lecture on this module, and it's been introduced because it's, in my opinion, it's a very, if not one of the most important emerging areas in molecular biology and molecular genetics. So it's only right that you guys get an introduction to it at this stage in the degree. So thus far, as I said, we've discussed classical non-coding RNAs, but over the last 5 years there's been an explosion really in research carried out on several new types of non-coding RNAs. Now, it turns out universally that these RNAs that have been identified, these new non-coding RNAs, are all involved in the regulation of protein production rather than the physical act of protein production. So let me just explain what I mean by that. So the RNAs that we've talked about already, transfer RNAs, tRNAs, ribosomal RNAs, SNO RNAs, the small nuclear RNAs, these are all RNAs that are very important in the production of protein. Yeah. So obviously the trans tRNAs, the ribosomal RNAs, and the SNO RNAs are all involved in the process of creating ribosomes and producing protein. Small nuclear RNAs they're important in the act of splicing to get the mature messenger RNA ready to be made into protein. What these new non-coding RNAs are involved in is a regulation of protein production. So not the actual production of protein itself, but how much is made. Yeah, in essence, or to put it another way, the control of gene expression levels. Yeah. And one of the really interesting things really about the discovery of these new types of RNA molecules, at least in higher organisms, is that the vast majority of the actual genome, as we understand it, certainly in humans, appears to be actually involved in regulating how the minority of the sequence is used to code for proteins. So let me explain that in another way, because I don't think that was very clear. For example, if we consider the fact that of our genome, only 1.4% of the actual nucleotides represent DNA sequence which goes on to code for protein, the rest, the other 98.6%, is effectively non-coding. Yeah, that 98.6%, rather than being just junk, as it was described for a long time, is actually largely involved in the regulation of the expression of this 1%. And it's interesting that that correlation between large genomes with small amounts of coding region correlates well with organism complexity. So if we have a look here at, for example, some these are different types of fungus and E. coli, down here bacteria. Let's use E. coli as an example. If you look at the genome of E. coli, 86% of the nucleotides within the genome of E. coli are effectively encode for protein. So 86% of the nucleotides within the E. coli genome are there because they encode for protein sequence that's going to be turned into protein, which is then going to go, the proteins then go on to carry out cellular functions. As we progress through organisms, that ratio decreases. So Drosophila, fruit flies, these nematode worms, plants, these are model organisms which we're going to discuss in the next lecture. As the complexity increases, so does the ratio between non-coding genomic information and coding genomic information, until we get down to us, arguably the most complex of organisms, where we see that only 1% of the actual DNA sequence in our genome is responsible for coding. The rest of it, well, what's it doing? That's been a question that everybody's been asking for 100 years, or certainly for many years in science, and only now we're starting to be able to potentially answer that question as our understanding of these regulatory RNAs increases. Another thing I want to highlight to you just really is to emphasise what I mean by this area being very explosive at the minute and important for modern biology. This is a list of publications on small non-coding RNAs. These publications don't include publications relating to ribosomal RNA and the classical RNAs we talked about already. These are publications just on this new family of RNAs. And as you can see, in the last 5 years really, there's been almost an exponential increase in the amount of research activity going on and involving these particular molecules, which is indicative of just how important they are in biology. Okay then, so this new family of non-coding RNAs, it can kind of broadly be broken down into two further categories: long non-coding RNAs and short non-coding RNAs. So we'll consider the long variety first. These were actually identified in a lot of cases after the short non-coding RNAs that we're going to discuss in a while. But these long non-coding RNAs are really interesting actually. One of the reasons they're interesting is that they actually can be considered the Higgs boson of modern molecular genetics. And what I mean by that is that if you look at the transcriptome of a cell, so in other words all the transcriptional activity which a cell is undergoing, or all the transcription that's going on within a cell, you'd be forgiven for thinking, and I would have agreed with you up until recently, that the vast majority of transcription that's underway in a cell is basically working towards a production of messenger RNA molecules. So the cell's interested in transcribing DNA to make mRNA. We're not considering ribosomal RNA at this point anymore. However, that's wrong. If you actually look at the transcriptome of a cell, the vast majority of actual transcription doesn't produce mRNA, it produces these long non-coding RNAs. Up until recently we didn't even know existed. So from that point of view, they can almost be considered the Higgs boson of like the transcriptional universe, if you like. So it's these long non-coding RNAs that are actually making up the vast bulk of transcription which goes on within a cell. So where on earth did all these long non-coding RNAs come from, and what do they do? Are they important for the cell or what? So the actual evolution of these long non-coding RNAs is a very controversial area. So there are two camps in essence. The first camp argue that long non-coding RNAs are simply transcriptional noise. Yeah? And the other camp, perhaps not surprisingly, argue that they're not transcriptional noise, they're functionally important. By transcriptional noise, I mean that they're just kind of the products of background non-specific transcription. Yeah? And there is evidence to support that hypothesis. There is evidence that backs that up. The two main pieces of evidence being that the actual expression of these long non-coding RNAs, whilst they're very abundant overall, individual long non-coding RNAs are only very weakly expressed. So they're driven from very weak promoters. Yeah. That kind of supports this idea that actually what's going on with these long non-coding RNAs is that transcription factors in RNA polymerase II are just non-specifically binding to parts of the genome at random and producing just random sections or random pieces of RNA which don't actually do anything. They're just byproducts of a slight inefficiency in how the transcriptional machinery binds to the DNA within our genomes. And that's also backed up by the fact that there's actually very poor conservation between species for these long non-coding RNAs. So if you look at long non-coding RNAs, for example, from humans, I look at the sequence of those long non-coding RNAs and then ask, is a similar long non-coding RNA sequence present in mouse? We'll find that often they're not. There's no cross-species homology. Now that's obviously very different to what we see for genes. If you look at a very important gene from humans, such as a gene involved in the cell cycle, or P53 or something like that, you will see that it's very highly conserved sequence-wise across species, sometimes right through to bacteria. Yeah, and that's because those genes are very functionally important. So if these long non-coding RNAs were similarly important, surely we'd expect to see similar conservation between the species. So they're the major arguments for lncRNAs being not important and just being a byproduct, being transcriptional noise. But there are intriguing bits of evidence which suggest that the other camp may actually be correct, that these things are important. For example, there's a startling correlation between organism complexity and long non-coding RNA abundance. So as the organism becomes more complex, the number of long non-coding RNAs that organism produces increases. Yeah. And people argue that this is a strong indicator that actually these lncRNAs are involved in generating genetic diversity and perhaps regulating expression of the genome, as we'll see in the next slide or two. Also, whilst there's very little conservation between species and the general expression of these lncRNAs is weak, if you look at the actual pattern of expression, in other words, the tissues in which certain lncRNAs are expressed, that is conserved between species, suggesting that there is some element of conservation of function as well. So where did they come from then? Well, there are several hypotheses which are non-mutually exclusive, so any of these might be correct, or they might all be correct, or just a combination might be correct. But general consensus is that they are derived from the mutation of a normal protein coding gene. So the gene is mutated in such a way that it's no longer encodes for an mRNA that's going to produce protein, but the mRNA is still produced and then goes on to be a non-coding RNA. Could be a chromosomal rearrangement, two different parts of a chromosome come together because of a translocation event or a recombination event to produce one of these lncRNAs. Could be a duplication event, so as we'll see in a while, there are also short non-coding RNAs. If several of these are duplicated, it could end up in the production of a long non-coding RNA that has a new type of function. And finally, a major source of mutation in genomes is transposition, transposon elements jumping around the genome and inserting themselves where they're not wanted. And it's possible that such a transposition event may have led to the production of some of the long non-coding RNAs that we see also. They're categorized into different categories depending on where they're found in the genome. Sense long non-coding RNAs overlap with the sense strand of a protein coding gene. So here the protein coding genes are purple and the non-coding RNAs are in green. Antisense long non-coding RNAs overlap with the antisense strand of a protein coding gene. Bidirectional RNAs are located on the opposite strand of a protein coding gene but are not antisense to that gene. Intronic long non-coding RNAs are derived completely from within an intronic region of a gene. Yeah. So in a similar way to the small nucleolar RNAs we talked about earlier on. And intergenic long non-coding RNA sequences are just located in a part of the genome which is not near anything to do with a protein coding loci, protein coding gene. So I guess the important question is, what do they do then, these things? So we're expressing a hell of a lot of them. The more complex the organism, the more copies, the larger the amount of long non-coding RNA that is present. What are they actually doing? So for the vast majority that we know exist, we have absolutely no idea. So this is why the field is so exciting and why there's so many publications relating to these things at the moment, because it's a new area. It's a completely novel area. People have only been doing progressive research into this for the last 2 or 3 years, and over the next 2 or 3 years I'm sure we're going to see a lot of big publications explaining just exactly how these things are operating and what they're doing. What we can say is that for those few long non-coding RNAs that we have got meaningful scientific data for, it appears that they're involved in the regulation of gene expression. And it's interesting that they're able to actually assert this regulation of gene expression via many different mechanisms. So we've got some examples here of how they actually do that, along with examples of actual long non-coding RNAs from humans which have been shown to function in this way. So initially, one of the ways that they can affect gene expression is via the regulation of chromatin structure. So we talked about this in the gene expression lecture, the idea that a eukaryotic genome is packaged into a higher order structure with histone proteins into this chromatin structure, and that you need to gain access to that chromatin in order to express your genes. Well, the exist long non-coding RNA has been shown to be able to regulate that access. It does that via a cis mechanism, but also other long non-coding RNAs have been shown to function via a trans mechanism. So cis and trans, regards to biology, molecular biology, 99% of the time, if it's a cis mechanism, it means it's direct. Trans means it acts via an adapter or via a secondary mechanism. So cis silencing, for example, of chromatin structure like exist does, the actual long non-coding RNA, which is here in green, directly modifies the actual coding DNA or histone proteins in such a way that gene expression is turned off. So for example, it may lead to the methylation of the DNA and therefore the silencing of gene expression from that gene. Trans-silencing, the long non-coding RNA might modify another protein which then itself is involved in the actual alteration of chromatin structure. So rather than the RNA modifying the chromatin structure directly, it modifies it via an interaction with a third party. The actual transcription of these long non-coding RNAs themselves can affect gene expression. So it's possible that if a long non-coding RNA, for example, is being expressed near a protein coding gene, then the expression of this long non-coding RNA can titrate the transcriptional machinery away from where it's required in the genome. Yeah. So this is a protein that this is a gene that you wish to express. However, expression of the long non-coding RNA is titrating the RNA polymerase II enzymes away from the actual coding gene, thus turning off gene expression. Yeah. Conversely, the long non-coding RNAs might recruit a repressor of transcription, which normally leads to a coding gene being turned off. When the long non-coding RNA is present, it sequesters that repressor, turning on expression of this protein coding gene. There are trans mechanisms as well, whereby long non-coding RNAs might directly interfere with or recruit transcription factors to protein coding genes to turn on and off gene expression. Similarly, they may interact with transcription factors and promote that factor's recruitment to a gene, or restrict to transcription factors localization to the site of transcription, to the nucleus. So that's an example. An example there is the NRON long non-coding RNA, which is able to prevent the localization of a transcription factor to the nucleus of the cell, therefore turning off gene transcription. And finally, these long non-coding RNAs have also been shown to be able to affect the translation of actual mRNA via interfering with mRNA transcripts, associating with them and directing these transcripts for degradation in a mechanism similar to what we're going to discuss in a moment about RNA interference. Importantly, we've already seen articles which have shown that expression of these long non-coding RNAs is involved in diseases ranging from cancer through to Alzheimer's. So certainly these long non-coding RNA molecules are important from a disease point of view, human disease point of view. It's just that we just don't understand enough about how they're working to really get a grasp on how much they are involved in the etiology of diseases. So that's long non-coding RNAs. The other side of the coin are these small non-coding RNAs. And really these RNAs are what are responsible for, I guess, the most recent conceptual leap in our understanding of molecular genetics. Certainly in our understanding of gene expression. So these small non-coding RNAs are between 20 and 30 nucleotides long. They were first identified actually in C. elegans. Again, we're going to talk more about model organisms in the next lecture. The first small non-coding RNA to be identified was called ln4, microRNA. But it was really the work of two guys, Fire and Bellow, around the turn of the millennia that led to a big leap forward in our understanding of how these small RNA molecules are working. Since the work of Fire and Bellow, this field has just completely exploded and really revolutionised modern molecular genetics. And these small non-coding RNAs are now implicated in everything from heterochromatin control to mRNA destabilisation and control of translation. So there come in several flavours. The first example really is, and something that's used a lot around the world in research labs, are these so-called short interfering RNAs. So these are importantly double-stranded RNA molecules between 21 and 23 nucleotides long. They're involved in a process called RNA interference, which we're going to talk about in a second. And these short interfering RNAs are generated from precursor long double-stranded RNA molecules. Yeah? So you start with a long double-stranded piece of RNA which is chopped up into these 20 to 23 nucleotide long short interfering RNA molecules. And these long double-stranded molecules are chopped up by an enzyme called dicer prior to these short molecules then entering this RNA interference pathway. The second type, and I want to talk about these back to back because they're similar in a lot of ways, is a type of regulatory RNA called microRNAs, which you may have encountered already, depending on how much extra reading you've done for the other modules you've taken. These, like short interfering RNAs, are double-stranded RNA molecules between 21 and 23 nucleotides long. They're also involved in RNA interference, but importantly, microRNAs are produced via a very different mechanism to short interfering RNAs. So microRNAs are transcribed, often by RNA polymerase II, initially as a primary microRNA. And this primary microRNA is processed in the nucleus of the cell by an enzyme called DROSHA. Once DROSHA has processed the primary microRNA, it then, like short interfering RNAs, enters this RNA interference pathway. So what is this interference pathway then? Well, this is summarised in the figure opposite. I don't want to go into a lot of detail about this now. If you want to do some extra reading about it, that's fine. There's lots of information in the core text, particularly in molecular biology of the gene on RNA interference. It's something you're going to cover in more detail next year, so it's not a bad idea to do some extra reading on this. But I just want to talk you through how this process works. You won't need to understand the process in any more detail than I'm about to explain to you now in this lecture for the assessments on the module. So the first thing to recognise is that siRNA and microRNAs are derived from very different starting points. So siRNA, like we've said, is derived from a double-stranded RNA molecule initially, which is chopped up by an enzyme called dicer to produce these short interfering RNAs. MicroRNAs initially are produced via transcription by RNA polymerase II, and they produce these primary microRNAs which are then processed by DROSHA prior to being exported into the cytoplasm, where then they too are chopped up by dicer and enter the RNA interference pathway. The major player in the RNA interference pathway is this multiprotein complex called RISC, or RNA-induced silencing complex. What RISC does is it takes these 21 to 23 nucleotide long double-stranded short RNA molecules and manipulates them in such a way that the strands are separated to expose the base pairing that's present within the short RNA. That base pair sequence is then used to scan for mRNAs which have got complementary sequence to the short interfering RNA or the microRNA. In the case of short interfering RNAs, the sequence complementarity is normally 100%, and often the complementary sequence is found within the coding region of the mRNA. When RISC recognises and binds to a target mRNA, the mRNA is then cleaved, turning off gene expression, preventing translation of the mRNA into protein. MicroRNA is slightly different. Here, the complementarity between the RISC microRNA complex and the target mRNA is not 100%, so you'd see there's a region here which doesn't bind with 100% complementarity to the target mRNA. Also, binding tends to occur in the 3' untranslated region of the mRNA molecule rather than the coding region of the mRNA molecule. And rather than directly cleaving the mRNA, what happens when microRNAs bind to mRNA targets is those mRNAs are targeted for degradation and placed into a subcytoplasmic body called a P body or processing body, where they're eventually chewed up. So both processes lead to the repression of gene expression, but via slightly different mechanisms. And we'll talk in a lot more detail again next year about these two processes. But as I've said, you need to understand to a level that I've just explained there in the lecture how these two processes work and how they differ. So finally then there's another group of small regulatory RNAs which are very recent again, like the long non-coding ones, and these are these so-called Pee-wee interacting RNAs or Pee-wee RNA molecules. These are slightly longer than the siRNA and the microRNA molecules, are about 24 to 30 nucleotides long, and it's not really clear as yet how they're produced, but we do know that the production is independent of DICER. So unlike microRNAs and siRNA, these Pee-wee RNAs don't require DICER in order to be correctly processed prior to entry into the RNAi pathway. They are dependent on RISC, so these Pee-wee RNAs do associate with the RISC complex. And importantly, in addition to interacting with the RISC complex, they also interact with a special family of proteins called Pee-wee proteins. We don't really understand how these piRNAs are functioning, these Pee-wee RNAs are functioning as yet. What little is understood about what they're doing points towards a possible role in the silencing of transposons during embryonic development and spermatogenesis. So it looks as though these Pee-wee RNAs are very important in turning off transposition events during the production of the gametes. But again, this is a very new area, and certainly you don't need to worry about any more detail with regards to these RNAs than what I've just covered there in this slide. So then, just to recap, again on this new section, we have these new wave of non-coding RNAs that really represent a critical emerging area of modern biology. The long non-coding RNAs represent the majority of cellular transcription, but we know next to nothing about the function as yet, if they possess a function at all. Some examples are around of long non-coding RNAs affecting gene expression, which they do via many different mechanisms, but for the vast majority of these molecules we don't yet know what they do in the cell. The short non-coding RNAs are really an evolutionary shift in our understanding of gene expression in eukaryotic cells, originated in work from C. elegans, but now informs research and biology really across the board. Short interfering RNAs, important molecules in viral defense. So this I didn't mention when I was talking about them. The origin of short interfering RNAs is believed to be a mechanism for defending against invasion by viruses, which often have double-stranded RNA genomes. So the idea is that DICER would chop those double-stranded RNA genomes up, use the short interfering RNA molecules that are produced in order to target virus mRNAs and switch off viral production and virus replication. MicroRNAs are very similar to siRNAs in how they function and how they look, but they originate from a very different starting point, and in fact microRNAs are very, very important key players in developmental processes and also in a range of human diseases, particularly cancer. Much more on those next year. And the final class of regulatory RNAs are these Pee-wee RNAs, which seem to be involved in preventing transposition events during embryogenesis and are produced via a different mechanism which we're not fully aware of as yet from microRNAs and short interfering RNAs, but do use the RISC complex. It's this RNAi machinery like those other two small RNAs, with the exception being that these RNAs interact with a particular family of proteins called Pee-wee proteins. So to summarise, that RNA biology is probably, arguably, the hottest area of molecular genetics that's currently around. I think if you talk to anybody who's at the cutting edge of molecular genetics at the moment, they'd be hard pressed to put up an argument against RNA biology being really at the forefront of what's going on, certainly with our understanding of gene expression. Regulatory RNAs, both long and small, effectively represent a really dizzying additional level of regulation over how we use the information in our 20,000 odd genes. So that's exemplified by all this evidence and these correlations between the amount of non-coding information in our genome and genome complexity, etc. And it kind of points towards an answer as to why our genomes are so large, yet so little of it is used to code genes. Yeah, so it may turn out that what's once referred to as junk is actually the reason that an organism as complex as our cells can be produced from such a small number of protein coding genes. So thanks for your attention. I know it's a complex area, this. There's an excellent chapter in the Cortex Molecular Biology of the Genes on regulatory RNA. I will put some extra reading up on Blackboard as well towards the end of the semester for you to look at. I hope this has proven useful, and be continuing to podcast the other lectures in molecular genetics as well. So look out for those on YouTube. Right, thank you.